For centuries, the Turin Shroud has been one of the most contested objects in human history. Some believe it is the burial cloth of Jesus of Nazareth. Others argue it is an elaborate medieval forgery. Between those two positions sits a long history of investigation, skepticism, belief, and controversy. But in recent decades, the debate shifted in a critical way. Science stopped asking what the shroud meant and started asking what it contained. Deep within the linen fibers of the shroud, researchers began finding biological material, not symbols, not interpretations, physical traces, DNA fragments, pollen grains, chemical residues, evidence that could be measured, cataloged, and tested. And when that evidence was analyzed, it created a problem, not just for believers, but for skeptics as well. Because what scientists discovered doesn't fit either narrative cleanly. The DNA tells a story that neither side expected. And once you understand what it shows, the debate becomes far more complicated than anyone wanted it to be. DNA was never expected to identify the man wrapped in the cloth. That was never the goal. The objective was simpler and far more revealing, to reconstruct the history of the cloth itself, where it had been, who had handled it, what environments it had passed through. To do that, scientists focused on mitochondrial DNA, genetic material passed down through maternal lines and far more resilient than nuclear DNA. It survives longer in ancient material and provides powerful clues about geographic origin. Using sterile microvacuum tools and ultra-clean laboratory conditions, researchers collected microscopic particles trapped deep between the linen threads. Not surface contamination, but material sealed into the fabric over long periods of time. The results were published in peer-reviewed scientific literature. And they weren't simple. If the shroud were a medieval forgery created in Europe, the genetic signal should have been dominated by Western, European DNA. If it were a relic that had remained in the Middle East, traces should have clustered around that region. Either outcome would have supported one side of the debate cleanly. That is not what appeared. The genetic material recovered from the shroud reflected multiple human populations across vast geographic regions. Middle Eastern lineages consistent with ancient Levantine populations. Western European DNA consistent with centuries of documented handling in France and Italy. But also, traces linked to North Africa, South Asia, and East Asia, including haplogroups associated with regions as far away as India and China. This was not the profile of a single individual. It was a biological record of movement, a genetic map of human contact spanning continents and centuries. And that immediately raised a profound problem for the forgery hypothesis. A medieval forger working in 14th century Europe could not realistically account for this pattern. In the 1300s, global travel was limited, slow, and extraordinarily rare. There was no mechanism for a single piece of cloth manufactured in Europe to accumulate genetic material from such a broad and geographically specific range of populations, unless it had actually passed through those regions over extended periods of time. Random contamination doesn't create structured geographic signals. History does, and the DNA wasn't showing random noise. It was showing a pattern that mapped directly onto documented trade routes and pilgrimage paths that connected the ancient Mediterranean world to Asia. When researchers mapped the DNA findings spatially, something unexpected emerged. The genetic distribution closely aligned with a documented, though often dismissed, historical movement of the shroud through the ancient world. Early records, some dating back to the 6th century, describe the cloth traveling from Jerusalem to Edessa, then to Constantinople, before arriving in Western Europe, centuries later during the Crusades. Edessa, in particular, sat directly on major Silk Road trade routes. It was a crossroads connecting the Mediterranean world with Persia, India, and East Asia. For centuries, pilgrims, merchants, diplomats, and clergy would have approached, touched, kissed, and handled the cloth. Religious relics in the medieval period weren't kept behind glass. They were venerated through physical contact. Each interaction left microscopic biological traces, layer upon layer, century after century. What the DNA appears to record is not a miracle. It's contact. Human contact across vast distances and deep time. The genetic signature is precisely what you would expect from an object that spent centuries moving through the ancient world's most important trade and pilgrimage networks. It's what you would not expect from a cloth made by a forger in medieval France. Independent confirmation came from palynology, the study of pollen. Researchers identified pollen grains from dozens of plant species embedded in the shroud's fibers. Some were European, expected. Others originated from Anatolia and the Middle East. Most strikingly, several species were native only to a narrow ecological corridor between Jerusalem and Jericho in the Judean desert. These weren't common species that could appear anywhere. They were endemic to a specific geographic zone. Pollen cannot be painted on. It cannot be artistically applied. It acts as a geographic fingerprint, locking an object to specific environments where those plants grew. The pollen didn't just show the shroud had been in the Middle East. It showed the shroud had been in very specific locations within the Middle East, locations that align with the documented path from Jerusalem through the Judean wilderness. Again, the pattern matched movement, not fabrication. It matched history, not hoax. Chemical analysis of the reddish stains visible on the shroud added another layer of constraint. Tests showed they were not pigments, they were human blood, and not the blood of a healthy individual. Researchers identified unusually high levels of bilirubin, creatinine, and ferritin, compounds associated with extreme trauma, severe muscle breakdown, and acute physiological stress. These biochemical signatures are consistent with prolonged physical abuse, dehydration, and shock. Bilirubin accumulates when red blood cells break down under extreme stress. Creatinine spikes during muscle tissue damage. Ferritin rises in response to severe trauma and blood loss. Together, these compounds paint a picture of someone who endured catastrophic physical suffering before death. These are not compounds you can fake with medieval painting techniques. They are not symbolic. They are physiological. They are the biochemical aftermath of torture. For decades, one piece of evidence seemed to settle the entire debate. In 1988, radiocarbon dating of a sample from the shroud placed it firmly in the medieval period between 1260 and 1390 AD. The announcement was treated as definitive proof of forgery, case closed. But decades later, chemists identified a critical flaw. The sample used for testing came from a heavily handled, repaired edge of the cloth, not from the main body of the fabric. Subsequent microscopic analysis revealed cotton fibers and dye compounds in the tested sample that were completely inconsistent with the linen structure of the shroud's main body. The sample was a patch, a medieval repair sewn onto the edge of the cloth after centuries of wear and damage. The radiocarbon test dated the repair, not the original cloth. Later studies using alternative techniques that bypass surface contamination, including X-ray diffraction and vibrational spectroscopy methods, placed the linen's material structure and degradation pattern closer to first-century textiles from the Eastern Mediterranean. Not proof, but constraint. The material science doesn't support a medieval origin for the linen itself. None of this confirms religious belief. Science does not authenticate theology. DNA cannot prove or disprove the resurrection. Blood chemistry cannot measure divinity. But science does impose limits on explanation. And the accumulated evidence makes one thing increasingly difficult to maintain, that the Turin Shroud is a simple medieval forgery created in isolation by a European artist. The cloth carries a complex, traceable history written at microscopic scale. It behaves less like an artwork and more like an object that moved, was handled, was venerated, and endured across centuries of human history, spanning multiple continents. The DNA doesn't tell us who was wrapped in the cloth, but it does tell us the cloth's journey was real, the contacts were real, the geographic movement was real. The shroud could still be a relic of someone other than Jesus. It could be the burial cloth of an unknown crucifixion victim from first-century Judea, whose cloth was later venerated as something it wasn't. That's possible. But what becomes increasingly implausible is the idea that it's a medieval fake. The evidence doesn't support that anymore. And that's what makes this so uncomfortable for everyone involved. Believers wanted science to prove it was Jesus. Skeptics wanted science to prove it was a forgery. Instead, science proved the cloth has a documented, traceable, multi-continental history that nobody can fully explain. It's real, it's ancient, it traveled, it was venerated. But whether it's the burial cloth of Jesus of Nazareth remains beyond the reach of empirical measurement. The shroud remains contested But the science has fundamentally changed the conversation. And once evidence enters the record, it doesn't leave quietly. History isn't settled by belief or by dismissal. It's measured. And the measurements keep pointing in directions neither side expected. Thanks for watching. If you enjoy how science rewrites what we thought, we knew about history's greatest mysteries, make sure to like and subscribe so you don't miss the next discovery. This is Hidden Strata,